want to start that now and then we can pause it. Sorry, I'm not trying to ignore you. Hi, what's up? We're just right in that office.
How you doing?
Were you getting worried? Pains?
Maybe.
About what?
That we hadn't arrived yet?
Oh, no, me worried? It's gotta have something to do with surgical ICU, to get my pulse rate up. Or a race. Really. Let me know when it's 2.
I still got like 5 minutes.
Yeah, we're good. I know, we know of at least 5 people who have lost. So. Have you seen a mirror image of this?
Over there. Yeah. We did notice that. It was big.
See them, they don't stay.
Yeah, I heard the story. Oh, yes.
I'm the one who has to walk there. The slide, screen goes up, and there's a shot. This stays in shot. We say, Tam, are they in the building? Or wouldn't be other way?
I'll be able to.
Escort them? Okay. Oh, awesome. Glad we held on. Maybe a few more.
Oh, come on with the freaking volume.
AI. Hope your PowerPoint hasn't been infiltrated. Didn't come from me.
There are 12 people.
Ah, here we go. Yeah. Who knew? I'm really glad someone else is in a white coat and scrubs, so that if anyone gets lightheaded in here, we send them to, not me. Oh, yeah.
I'm surprised you are not.
I didn't have any clean ones. All right. I don't see a, yeah. Okay. All right, with that emphatic door slam, I think it looks like showtime. So it's my great privilege today to introduce our speaker and let you hear her action-packed and very timely story. I think the best way to do this quickly is to get out of her way, is to say a few words about her topic in historical perspective. According to the genius who brought us Huck Finn, history doesn't repeat itself, but it often rhymes. Exactly a century ago, Paris hosted the Olympics. Swimming in the Seine was newly prohibited. The responsible bug, Escherichia, i.e. E. coli, had just been named after its German discoverer, and the news of the British track team's dominance solely disseminated without the benefit of NBC, X, or TikTok. But you know, for most fans, the backstory on what happened then had to wait at least 50 years for Chariots of the Fire, the big blockbuster biopic. But for readers of Nature, a much more insightful coverage of the story behind the story appeared in something a lot closer to real time. And that is this, except it's not responding. There you go. So notice this, 1923, 24, 25. A year earlier, Hill had made an arguably most important discovery of his life. And that was the N of 1 experiment that proved to be generalizable to all vertebrate species. The N of 1 was himself, as I think you can see right in here, H. And after breathing into an airtight bag at various running speeds around a quarter mile around the track, in Hill's words, the oxygen intake rises steadily as the speed is increased, attaining a maximum beyond which no bodily effort can drive it. Well, that turned out to be the concept of VO2 max and later the lactate threshold, which every Olympiad that made it to Paris this go round has had all this testing 100 years on. So he's about 100 years ahead of his time, along with Penn professor who he shared the Nobel Prize with a year earlier, Otto Meyerhoff, who I think finished out the work on, Mike, you could correct me if I'm wrong on this, finished out his work on the on glycolysis within 100 feet of where we're sitting right now, somewhere in this building. Yep. When asked what on earth led him to become so obsessed with the topic, Hill's answer was that it related to his personal experiences running track, where he posted very impressive times in essentially all the individual events, but was never able to find a single event where he could earn an international podium. Instead, his consulting input proved invaluable to the British track team 100 years ago and all subsequent athletes that achieved international rank, not to mention countless patients everywhere undergoing cardiopulmonary exercise testing. All of this is to say that Coral reminds me of the best historical accounts of A.V. Hill. Her thesis not only directly builds on the classical studies of Hill and his protege Katz, who 30 years later also won a Nobel Prize, but as you'll see, her conceptual approach has been influenced by her side hustle as a member of the U.S. rowing team. I can assure you that the entire lab shared her dreams of a podium finish this week in Paris, but today I'd like to welcome her to a podium of arguably greater significance as she defends an incredible body of research. She's contributed to so many other projects that have been the subject of her other presentations. For instance, she'll make only brief and passing mention at the end of the subject of the papers in her seminar at the last meeting of the ASGCT. Today, she focuses almost entirely on the very timely topic of genetic cardiomyopathy and muscular dystrophy. The subject was a major focus of discussion at that gene therapy meeting as two related major news stories broke out on opening day. We desperately need better primary outcome measures, and these must be built on sound basic translational and clinical science. In the touchy FDA-regulated business of extrapolating from mouse to man, much comes down to epistemology and the often subtle distinction between can't and want. Start with vexing questions like, why do even dystrophic mice want to run 2 miles if given access to a running? Food's over there, the water's over there. They get in the running and they just go. In closing, I'm delighted to say that Coral has earned the privilege of presenting an amazing body of work made possible by a larger team, most of whom she trained to do all the complex and labor-intense assays to the highest standards of scientific rigor. The best and rarest of news for all of us is that only hours ago, we got formal notice of an NIH translational grant designed to propel all of this team's wet bench work to the bedside as soon as possible. Hopefully, Coral's work will directly help patients suffering from the most common single gene lethal in man. Coral, in great appreciation, here's your podium.
Oh, thank you. Oh, oh, thank you. Oh, hey. I didn't, sorry. No, that was all him. All right, well, thank you so much for that introduction, and thank you all so much for being here. I'm very excited to share with you what I've been working on for six years. A lot of this data actually came in the last 2 to 3 as a PhD normally occurs. So I am in the gene therapy and vaccines program, and I've been really privileged to make a COVID vaccine as well as work on a gene therapy while I've been here. So it's been, I feel like, a really productive PhD. So first, I'm going to run through some basic concepts just so we can really understand the bulk of my thesis, and then we'll dive right into the thesis. And we'll finish with some Q&A and extended acknowledgements. So me and my lab are trying to cure Duchenne muscular dystrophy. And I'm going to be presenting a lot of mouse data today, so I wanted to use this slide to really reconnect us back to who we are trying to help. As researchers, we don't get to see the faces of the people that we are trying to help, mostly because what we do today does not show up in the clinic for 10 to 30 years. What I'm about to show you has been built off of the last 30 years of work. And our lab is in a really exciting position because, just like Hansel mentioned, we finally have the opportunity to go into clinical trials and treat human patients within the next 2 to 3 years. So Duchenne muscular dystrophy is a childhood onset muscle wasting disease and impacts 1 in 4,000 male births worldwide. Symptoms show up at about 2 to 3 years of age, starting with motor function delay. By 5 years, you see a more pronounced reduction in walking ability. And then by 5th grade, you have complete loss of lower limb ambulation, leading to full-time wheelchair. By high school is when respiratory impairment starts to show up and you are put on a ventilator. And by your late 20s is when your heart starts to fail, usually due to dilated cardiomyopathy. You'll lose heart function and ultimately succumb to the disease through that. This is a genetic disease, so these children are born with it. It is X-linked recessive, meaning that it predominantly impacts males. This is because they only have 1 X chromosome, so if they have 1 defective X, they do not have a functional X to compensate in a lot of their cells. This disease is caused by mutations in the dystrophin gene that result in no dystrophin protein. So for the next few slides, I'm going to cover how a genetic disease manifests as a full body disease. So let's start at the cellular level with the central dogma. So here I'm showing the X chromosome. And on that X chromosome is the healthy dystrophin gene. Now our own cell machinery is going to transcribe that DNA sequence into RNA, translate it into a peptide, and then it's going to fold properly to form the correct dystrophin protein that is functional. However, in DMD, you have a mutated dystrophin gene, shown here as like a huge deletion that leads to a mutated RNA, mutated peptide that's not going to fold properly or be super truncated. And now we're not going to get any dystrophin protein. So the reason why the dystrophin gene is so affected is it is one of the largest genes in the human genome at about 2.2 million base pairs. So that makes it more prone to mutation during replication. About 70% of these are large deletions, 20% are duplications, and the rest are point mutations that lead to premature stop codons. All of these mutations result in a disrupted reading frame. So just real briefly, we're gonna look at exon 51 in the dystrophin gene. I'm showing the DNA sequence here, and the way the DNA or RNA sequence is read is in these groups of 3 base pairs. Each one of those is going to code for a very specific amino acid that makes up the protein. When we have a deletion that shifts the reading frame, we're gonna move out of that triplicate reading. And so now we're gonna start our next codon with this AT, TA, and we're gonna pick up this CG from the glutamate. So it's really going to look like this, and just notice that this DNA sequence down here is different than the DNA sequence of the wild type. When that gets transcribed to RNA, this could result in a premature stop codon. So this is going to tell the ribosome to stop right here, do not make the rest of the protein. Alternatively, we'll just make a bunch of peptides or the rest of amino acids that are not in the wild type sequence, so we're not even making the real dystrophin protein. Okay. So what is this dystrophin protein even doing, and where is it? Now I'm gonna be talking about the muscle isoform of dystrophin. So we're gonna take the whole muscle up here, take like a little cross section, look down towards the tendon, and you can see that we have these fascicles that are filled with muscle cells. A muscle cell is a muscle fiber. Now, the muscle cell is highly specialized in that it has these bundles within it that are called myofibrils. These are essentially bundles of sarcomeres. This is a single sarcomere or contractile unit, and this is where the force is generated during muscle contraction. So when the pink myosin starts to pull the purple actin, those 2 Z discs here on the side are going to get closer, causing a shortening of the muscle and contraction. Now, how do we get all that force that's generated down here deep within the myofibrils out of the muscle cell down to the tendon and the bone to actually do work? That's where dystrophin comes in. So here we have the muscle cell, and we are going to zoom in on the membrane. So here is that membrane. We also call it the sarcolemma, and here is the dystrophin protein. Dystrophin is going to bind the intracellular actin down here at one end. That actin is going to be connected to the Z disc of the sarcomere. Then at the other end, it's going to bind this huge protein complex in the membrane. That membrane complex, transmembrane complex, is connected to the extracellular matrix. So dystrophin is serving as this mechanical linkage between the sarcomere and the outside world. And we think it is functioning by transmitting that force that's generated in the sarcomere up across dystrophin and safely through that membrane complex. Now, when we don't have dystrophin, we don't have that safe conduit through which the force can travel. And so upon subsequent contractions, we get muscle membrane weakening and eventually membrane rupture. That's going to lead to an influx of calcium, protease activation, and eventually death of that fiber or a segment of the fiber. So now, big picture, what's happening to the organ, we're going to get muscle membrane rupture, which is going to lead to death of that fiber. Then we're going to have muscle stem cells called satellite cells come in and regenerate that fiber. But remember, all of these fibers do not have any dystrophin. So we're actually going to get this concomitant process of regeneration and degeneration happening at the same time. At about age 5 to 7, you're going to blow through all of your stem cell reserves. And now you're going to get replacement with fibrosis and fatty tissue. And that's what's going to lead to loss of ambulation. So the best current treatment for the symptoms right now are corticosteroids. They reduce inflammation, improve muscle membrane repair, and preserve muscle mass and strength. If you take these consistently for 1 year, you can push back disease milestones by up to 2 years in your legs and up to 8 years in your upper body. So this has been huge. The next drug that is FDA approved, it is an attempt to cure, well, not quite cure, but at least Beckerize this, meaning we're going to make it a milder version, is an exon skipping drug. And notice how there are multiple infusion bags because you have to take this weekly or monthly for the rest of your life. Briefly, the way this works, remember how we disrupted the reading frame. I'm going to show exon 51. Here we have that point, we have a mutation that disrupts the reading frame. So notice how exon 52 is blurred out. We're not making the rest of this protein. What the exon skipping drugs are these little antisense oligos that are going to tell the cell to skip over the mutated exon or region and link up again with the healthy exon 52. So we are going to truncate the protein, but we are maintaining the reading frame, so we do get some version of a dystrophin protein. The only issue with this is that we've only been able to increase dystrophin expression by about 1 to 5% of wild type levels. So it really has not been as beneficial as we'd hoped. The therapy I'm going to talk to you today about is gene therapy. Notice how there's only one infusion bag because theoretically you only need one shot and done. There are pros and cons to that, and I'll get into that. So the way gene therapy works is you still have your mutated process going on in the background. We're not editing your genome, but we are delivering the correct version of that gene exogenously or from the outside. So we're going to deliver the correct version. It's going to sit next to your genomic DNA. We call that the transgene. Your own cell's machinery is then going to transcribe that into RNA, translate it into protein, and now you're going to make the functional version. The gene therapy components are the adeno-associated virus as our delivery vector and a transcriptional cassette. That's going to contain our therapeutic transgene and then a promoter to turn it on in target tissues. Now, right now we're using AAV because it is non-pathogenic, so it cannot replicate on its own. And there are various serotypes that confer various tissue tropisms, so we can more selectively target what cells we're getting into. Lastly, it does confer stable expression in non-dividing cells, which is that one shot and done. We can deliver this to our non-dividing skeletal muscle, and it should sit next to our genomic DNA, making that correct protein for the rest of our life or that cell's life. Now, the way this works is we're going to take the virus's own DNA, we're going to gut it, and we're going to replace it with our therapeutic transcriptional cassette. Then this cassette is going to be packaged into AAV that we can deliver systemically. One limitation specifically with DMD gene therapy is the size we have to work with. This virus can only hold about 5,000 base pairs, but the dystrophin coding sequence is 11,000. On top of that, we have to include a promoter and regulatory elements. So the field has had to find ways to strategically truncate the coding sequence to make a truncated protein that still somehow preserves the function. And this is what that transgene is going to look like, but now we can at least package it into AAV and deliver it systemically. These are the current constructs in clinical trials right now. The big takeaway is that these are the ways that they have truncated it. We've taken away this huge chunk of the rod domain and we've chopped off the C terminus so that we can fit into AAV. These are all derived from dystrophin. Now, some of the headlines recently, this happened just this May, Pfizer reported its second death that was treatment related. This kid was 4 years old and he died from cardiac arrest, which was very weird because usually a cardiomyopathy does not occur until later in the disease progression. So they're investigating this right now. Unfortunately, about a month later, Pfizer reported its phase 3 results and they completely missed their primary endpoints, which was to improve motor function. Now, Sarepta is the furthest in the game. They received accelerated FDA approval only for 4 to 5-year-olds just last year. But then a few months after they got that accelerated approval, they also reported that they failed to meet their primary endpoint and they did not improve any motor function. But they still sought to expand their approval so they can inject more than just 4 to 5-year-olds. And just about a month ago, they received that expanded approval to inject all kids 4 and above with Duchenne. So another limitation in really any gene transfer therapy is that it can be immunogenic. Remember, DMD patients do not make any dystrophin protein, and so their immune system has not been educated to recognize these dystrophin peptides as self. Also, if you have a large deletion, then you're missing a large region that can't be expressed in the thymus, so your T cells can't even be educated to that. All current gene therapies for DMD are delivering some form of dystrophin. So there is the potential for the immune system to attack those cells that are transduced with our therapy. And we've seen this in multiple patients across many studies. I'm going to show you one from Sarepta. So this patient presented with immune-mediated myositis. So immune system was attacking his own muscle. So they looked at his T cells These are T cells specifically reactive to microdystrophin, which is the gene therapy. And you can see that he had high T cell reactivity to the gene therapy that persisted 1 year and was still detectable after 2. So the big takeaway from this is that it is a start. The disease burden is huge and we need to start somewhere, but we're not there just because we have an FDA approval. So we do need to continue to improve. So that moves us to what my lab is working on. We want to replace dystrophin with the protein called utrophin. It is a non-immunogenic alternative that is structurally, functionally, and evolutionarily related to dystrophin. And I have these mock 3D renderings of both proteins, so they do have very similar structure and function. Now, utrophin is also a muscle protein. It is expressed at myotendinous junctions and at neuromuscular junctions where the neurons synapse with the muscle. So here, utrophin at the myotendinous junction, we are still binding that protein complex at the membrane and binding actin at the other side. At the neuromuscular junction, again, complex in the membrane, binding actin at the other side, and potentially serving as a scaffold for other signaling receptors like these acetylcholine receptors. So utrophin is not mutated in DMD, so again, also recognized as self by the immune system, and it's already been shown in transgenic animals that we can fully replace dystrophin with utrophin. So we're going to take this into our gene therapy design. However, the coding sequence is 10,000 base pairs. So we had to truncate it as well. And we took out this huge region of the rod domain and chopped the C terminus. Now, we've already published that it works in skeletal muscle. So I'm just going to go through this briefly. The format's a little odd, that's okay. So here we have our untreated DMD mouse, and you can see it's highly inflammatory with mononuclear cell infiltrates, and we also have some centrally nucleated cells. When the nuclei migrate into the center of the fiber, that means the fiber is regenerating. We don't want that. Whereas here with our treated DMD mouse muscle, we have peripherally nucleated cells and no inflammation. I think the biggest experiment from this paper was showing that it was non-immunogenic. So they took dystrophin null dogs and they injected one leg with AAV microutrophin and the other leg with AAV microdystrophin. Then over time, they took the T cells and again, they looked at T cell reactivity, and there was high T cell reactivity to microdystrophin peptide pools, but in the same animal, undetectable reactivity to microutrophin peptide pools. And so, okay, my crops did not transfer, but I was just showing that it was very inflammatory. The AAV microdystrophin was highly inflammatory in the leg that was injected with microdystrophin. So a big takeaway, the benefits of using microutrophin is that it can functionally replace dystrophin and it is non-immunogenic. So this leads me to my thesis project. We showed it restores the skeletal muscle, but does it actually protect the heart? So dystrophin deficiency in the heart is very similar to skeletal muscle in that it does start with weakened membranes that will ultimately rupture and lead to cardiomyocyte cell death. However, there's not the same regenerative capacity in the heart as the skeletal muscle, and so we do get greater levels of fibrotic replacement. After that, that fibrosis is very poor at contracting, and so it does start to thin and stretch, and that's where we get that dilated cardiomyopathy, where you have a dilated left ventricle that loses function. And eventually, with that lost function, you'll experience heart failure and succumb to the disease. Unfortunately, our DMD mouse model that we call MDX does not show an early dilated cardiomyopathy. So the field has had to strategically use drugs to accelerate injury in the MDX heart in order to even distinguish MDX from wild type. So the way it works is we will use a drug to increase heart rate and the strength of contraction, which will lead to membrane weakening in the MDX membranes. But if you have dystrophin and you have this link between the cytoskeleton and the outside world, your membrane is protected. And if you don't, then that increased cardiac workload is going to result in weakened membranes and rupture. So we usually use the drug isoproterenol, which stimulates the sympathetic nervous system. So that's the fight or flight. And the way it works is iso isoproterenol, or I'm going to call it ISO from now on, is going to bind receptors on the cardiomyocytes, activate downstream signaling cascades, which is going to increase intracellular calcium levels that are going to bind this along the sarcomere and activate heart rate and contractility. That increased contractility is going to accelerate fiber damage in the MDX heart, but not in our wild type. The only issue is that there has been high variability in outcome measures with ISO in MDX. Some doses have been very lethal, and yet if you multiply that by 20, the MDX are fine for a month. So part of my thesis was to develop a pharmacologic assay that was actually consistent and reproducible. So we're still going to use isoproterenol, but we're actually going to add another drug to that, which is called atropine. Atropine does signal through a lot of different pathways, but the one that I found really cool and used in this project was that atropine can actually enhance the effects of ISO by entering cells through cation channels like OCT3. It'll bind phosphodiesterase 4D. PDE4 binds cyclic AMP and degrades it. But when atropine binds PDE4, it prevents it from degrading cAMP. So now we have high cytosolic levels of cyclic AMP, which then can bind all of these downstream proteins, which will lead to an increase in intracellular calcium and increased heart rate and contractility. What's really cool is that it'll elevate the heart rate even more than ISO alone and for a longer duration, which should increase injury in the MDX mouse. So taken together, we increase heart rate and contraction with ISO and atropine, which will prolong the cardiac workload, injure MDX. Now we're going to test our gene therapy. So we do need readouts to see if we are actually injuring our mice. And so we are going to use blood cardiac troponin I, Evans blue dye, and exercise assessment and cardiac remodeling. Cardiac troponin I is one of these small regulatory proteins in the sarcomere. Remember this down here in the myofibrils. When the muscle membrane is ruptured, that cardiac troponin I is going to leak out systemically. So now we can measure that in the blood. We should never have cardiac troponin I in our blood. So this is a highly specific biomarker. There is also no cross-reactivity to troponin I in skeletal muscle. This is because cardiac troponin I has this unique epitope that is exclusive to it that is not found in skeletal muscle troponins. I am gonna hone in on this because I am gonna be doing exercise readouts. So I do wanna make it clear, this is gonna be cardiac specific injury, no cross-reactivity with skeletal muscle troponin, which can be highly specific. Next is Evans blue dye. This is a blue dye and we'll inject it systemically and it'll do the opposite. It'll get into injured muscle fibers and light up the whole fiber blue, which will fluoresce like a red magenta. So any signal indicates that this whole region or that all those fibers have been damaged. So this was the initial experimental setup. We injected with either atropine or saline, so with or without atropine, and ISO. Then from there, we divided our mice into two groups. One was received a terminal bleed by cardiac puncture so that we could get some blood and measure cardiac troponin I. The next group was injected with Evans blue dye immediately after the challenge. Then we let the Evans blue dye circulate overnight and then we euthanized for histology. So first I'm going to show you the ISO only without atropine. This is our wild type healthy. This is our diseased MDX mouse. And you can see by Evans blue dye, we do have these large patches of injury, which is good. However, when we add atropine, you can see from the back of the room, this poor mouse accrued a massive amount of injury by Evans blue dye. And when we quantify it here, you can see that we have a significant increase in our MDX mouse and really no difference without atropine. And these are the troponin levels. Again, we have a significant increase in troponin levels as well, and in a very good scale from about 1 to 15, whereas without atropine, we're only going from about 0.5 to 1.5. So before we test our gene therapy in this experiment, we want to make sure that our gene therapy is actually expressing in the heart. So we injected neonates at about 10 days of age systemically, and then we aged them 5 months. Then we looked at expression. So in the green is eutrophin. So you can see that there is very low signal in our MDX mouse, and then we get robust signal in our treated animals, and we're hitting nearly all myofibers. This is a WGA stain, wheat germ agglutinin. It just binds extracellular matrix around the fibers just so we can see where the fibers are. Then we look to see if it recruited that protein complex in the membrane. So we're gonna use one of the proteins in that complex called gamma sarcoglycan as a surrogate for the whole complex. And you can see in wild type, where we have full length dystrophin, we are recruiting gamma sarc, our MDX is not, and then our treated, we have a robust expression and co-localization to the membrane. Now we're gonna test them in this assay. So we injected at 10 days, we aged them 5 months, and then we gave all mice atropine and iso. 2 hours later, we bled them for troponin, and then we injected Evans blue dye. By doing it this way and bleeding by retroorbital route, which is non-lethal, we're able to look at troponin and EBD in the same animal now. So here's the Evans blue dye. And again, MDX is getting hit massively. But what's really cool is that our microeutrophin treated has essentially no Evans blue dye. We are rescuing to a wild type response. And then by cardiac troponin, again, we see a significant reduction in troponin, indicating that our gene therapy is protective against acute cardiac injury. So now I wanted to go to something more physiologically relevant. So I want to know if it's protective during exercise, specifically in the heart. So it's been shown that if you have skeletal muscle, if you have healthy skeletal muscle, it increases your capacity for physical activity. If you do perform more physical activity, it's going to stress your heart even more with that cardiac workload. So it's been previously shown in transgenic mice. These are mice that have skeletal muscle dystrophin, so their skeletal muscle is healthy, but their heart does not have dystrophin. That they will run further distances than mice that don't have dystrophin in their skeletal muscle. However, this puts a huge strain on their heart that does not have dystrophin and leads to poor or even worsened cardiac outcomes. So I wanted to make sure, since we are improving the skeletal muscle, we've already published that it does improve running, is that improved physical activity actually protected in the heart? So it's very hard working with a disease animal because there are so many components to the disease. So MDX running performance is always impaired. It's always going to run less. And that's because it has skeletal muscle disease, cardiorespiratory disease, and its vasculature is all messed up. So I did consider using cardiac troponin I after running just to see what the actual cardiac injury after exercise was. Unfortunately, MDX mice don't run hard enough or long enough in a single bout of exercise to induce significant cardiac stress. So I had to think outside the box. I'm going to show you what happens in the troponins when they run. This is wild type. This is MDX. R is the run. So after a run, you do see a little blip in the MDX, but nothing to the degree of this atro-iso. So I wanted to develop a method that would test whether our microeutrophin in the heart contributes to exercise performance, but I had to do it in a way where the MDX skeletal muscle was not a limiting factor. So me and a postdoc, Maria, we developed this dual drug and exercise stress test. So first, we're going to give them that atropine iso drug challenge. This is going to selectively target and stress the heart. Then we're going to follow with an exercise assessment and see how their exercise performance does compared to baseline when they did not receive a cardiac stressor prior. Our two readouts are going to be the distance that they traveled during that test, as well as cardiac troponin I. And remember, this is cardiac specific, so it's going to be independent of skeletal muscle injury, even though they are running. So we first injected and then we aged the mice. I did do 5-month-olds and 10-month-olds, so I'm just going to show you the 10-month-old data today. At 10 months of age, we did take baseline levels of cardiac troponin I, and then we did after acclimating them to the treadmill, we did a baseline running performance. So this is the protocol, and I developed this protocol so that most MDX mice can complete the easy and manageable speeds but they were challenged in the last 5 minutes. So about 50% of MDX mice can complete the entire thing at baseline. 70% can complete this region. So now I found where their skeletal muscle limitation was. We did have removal criteria. So if they remained in the last 4 inches of the treadmill and received 3 consecutive shocks, they were removed. Or if through the whole 20 minutes they received 20 shocks, we removed them. We also had 2 blinded observers just to make sure that we were pulling mice appropriately. We also had an exclusion criteria. So MDX mice have a fear response. This is because there is a dystrophin isoform that's actually in the brain that increase, and so when you don't have dystrophin in the brain, you increase anxiety. So they will get very anxious just putting them in the treadmill no matter how much you acclimate them. And so if a mouse does not complete the first 5 minutes, we exclude them and all of their measurements from the study. All right, so we got our baseline. Then on a separate day, this is a whole single day challenge. We inject them with atro and iso. 2 hours later, we get the CTNI from the drug injury. Then we do the running assessment to see how well they perform after having a cardiac stressor. And then we will take final CTNIs after the dual drug and run. After that, those same animals are then going to run every single day for 20 minutes. And this is to look at long-term cardiac remodeling in response to daily exercise. So first I'm going to show you the running data. So we're going to look at our baseline run and our post-atro iso run. So here's our wild type. They were able to complete it at baseline and after the challenge, but our MDX mice experienced a severe reduction in total distance traveled. What was really cool is that our microutrophin actually preserved their running performance throughout the challenge. And this is graphing their percent change from baseline. So you can see that the MDX dropped 25% of their total distance from their baseline run, but microutrophin preserved. So now we're going to look at cardiac troponin I, the cardiac specific injury. We've got our baseline, post-atro iso, and after the run as well. So first we're going to look at the atro iso, and again, even at 10 months post-treatment, we still get a reduction in cardiac troponin I after the cardiac stressor, which is great to see. But what's even cooler is that after the run, the MDX mice experienced a severe elevation in cardiac troponin I levels, whereas our microutrophin, again, maintained very low levels, which was really cool to see. What's also cool is that all of these mice ran less distance, so they had less of that exercise cardiac stressor, and yet they still had a lot of injury, indicating that the cardiac stressor or that cardiac stress was impacting their exercise performance, but our microutrophin in the heart was able to protect against that. I also wanted to look to make sure that cardiac troponin I didn't just naturally elevate over time. So I did have no run controls. These mice received atropine and iso, but they just did not run, and they were bled at the same time points. So here we have our wild type, and here we have our MDX, and you can see that the levels just taper off or go down. Now I'm going to look at cardiac remodeling in response to daily exercise. So again, all these mice went through this challenge. So these same mice are now going to just be run every day through that 20-minute protocol for 2 weeks. So cardiac remodeling is a physiologic, which is normal, or pathologic adaptation to some stimulus. Our 2 remodeling processes we're going to look at are cardiac hypertrophy and cardiac fibrosis. So hypertrophy in general is muscle fiber growth. If you experience concentric hypertrophy, your diameter of your fibers are going to get huge, they're going to get thicker. Whereas eccentric hypertrophy, your fiber is going to elongate. So we're looking at the cross section of a heart. Here's a normal left ventricle. This is the myocardium here. Concentric hypertrophy, if your diameter of your cells get bigger, your wall is going to get thicker. If your fibers elongate, it's going to increase this inner diameter of the left ventricle. Both are hypertrophy. So the normal physiologic stimuli for each, concentric hypertrophy is going to be a pressure overload. So that's going to be in response to increased blood pressure or increased systemic vascular resistance. You will usually get this from chronic strength training. like Jessica Butner, who has the world record, almost 600-pound deadlift. Eccentric contraction is going to be a volume overload. So this is going to be chronic endurance training, like Fiona O'Keefe, who made the 2024 Olympics in the marathon. So we're going to look at hypertrophy after the daily running. Here is our wild type MDX microutrophin. You can see the MDX mice do experience overall cardiac hypertrophy, and we measure that by heart rate normalized to their tibia length. But our microutrophin prevents cardiac hypertrophy. When I was taking pictures, I noticed that the fibers in the MDX heart were huge, noticeably huge compared to wild type and utrophin. So me and our lab tech at the time, Elaine, we sat there blinded and we traced 400 fibers per heart in all of our mice. And this is what we came up with. Concentric cardiac hypertrophy. Our MDX mice had huge myocyte cross-sectional areas indicating concentric cardiac hypertrophy, but our microutrophin treated prevented. But what's really interesting is that you would think that with running, you should get eccentric hypertrophy. But instead, we're getting concentric, which is more indicative of a pressure overload. I don't have time to go into this, but this was really cool to see. To me, this indicates that this response was in fact pathological or maladaptive, and that our microutrophin treatment prevented that. And just to show our no-run controls, again, these mice received atro-iso but didn't run, and then they did not run those 2 weeks. They just sat in their cage, shown here. And there's a moderate elevation, but nothing significant above baseline hypertrophy. But you can see here that the MDX mice that did run every day experienced significant increase in cardiac hypertrophy. And what's great is our microeutrophins are right in line with baseline MDX and wild type. We next looked at fibrosis. So this is a picroserous red stain. So the red is going to be fibrosis, and then the green is just a counter stain, so we can see the myocardium. And we did, in fact, reduce fibrosis significantly. We didn't quite rescue to wild type levels, but this is a huge improvement. I did want to look at whether fibrosis was being impacted by the daily run, or was it from the initial atro, iso, and run acute injury. Usually, if you have an acute injury that leads to myocyte cell death, it can be replaced by fibrosis. So I wasn't sure if it was running 2 weeks or that initial impact. And here you can see that in the no run, they are actually elevated from baseline. We didn't quite reach significance, but it's trending that way. What's very interesting is that these no runs did not have as high of a CTNI spike as our mice that did run, which is demonstrated here. So then I looked at the correlation between the initial acute injury, atro-iso run from that single day challenge against fibrosis 2 weeks later. And there was a significant correlation. So if you had a high troponin, you had high fibrosis. And all of our groups kind of segregate nicely along the linear trend line. So we have all of our MDX out here if you had high troponin, high fibrosis, then our no run controls, and then our microutrophin and our wild types are segregating nicely at the low end. However, I was a little concerned about these two mice that had significantly higher fibrosis. It turns out these two mice are actually these two mice who had low cardiac troponin, meaning that their injury was not inducing the fibrotic response we're seeing. So then I looked at microutrophin protein expression levels just to see maybe they don't have enough microutrophin to protect them. And it turns out it is true. So this is Western blot data. So we're looking at protein expression in the green. So if you have higher protein expression, you have lower fibrosis, which is in purple. Whereas these two mice had 50 to 75% less microutrophin expression, and yet they had more fibrosis. So all of this together indicates that if you have microutrophin, especially hitting a threshold level, you are protective against acute injury and fibrosis. Lastly, I looked at the relationship between hypertrophy and fibrosis. So hypertrophy can be physiologic when it is reversible. It is pathologic when it's irreversible and accompanied by fibrosis. So I wanted to see, was it accompanied by fibrosis? And there was in fact a relationship. So if you had high hypertrophy, you had higher fibrosis, indicating this was a pathological response. And again, most of our microutrophin treated are down here with wild type. So our major takeaways was that I developed a reproducible drug and exercise stress model, and our AAV microutrophin treatment is durable and supports increased cardiac workload through daily exercise. In the pipeline, we are testing next generation transgene designs for DMD to increase the stability of our protein product, and we're about to go into an IND enabling study. And with that, I'd like to thank my whole lab, Hansel, GTV, and all of you. And I'll open it up for questions. Yes, in the back. Thank you.
What's the stability of expression of time? You're looking 10 months out compared to like the injection set 10 days. So with the AAV therapy, is there a deep loss of AAV genomes over time? How stable is this, say, 1 dose?
Yeah, that's a great question. So the question, oh,
What? There's still an effect, but is there a change in transgene expression?
I see. Okay, so the stability of the treatment, we do see maintained vector genome copies. So at 5 months, I'm seeing anywhere from 1 to even 12 at our dose per diploid genome. And then at 9 months, I'm seeing about 1 to maybe 10.
Earlier on, where you if you look at the 1 month or shortly after.
That is a great question. I don't have data on that at this dose that we used. I've noticed in other tissues like the diaphragm, we'll get moderate transduction with AAV9 at about 3 weeks, and then we don't see anything after that. But not in the heart. Yeah, thank you. Yes.
Assume that full protein would be more effective as like same amounts of user working better or
That is a really good question. The reason, let me see if I can do this. I want to show you a Western blot because you are right. The full length is probably going to be more beneficial. We haven't found a way to actually increase endogenous utrophin to therapeutic levels, which has been a huge limitation. Microutrophin has been working great. However, we see this fragment band on our Western blot. So in all first generation, so that's the microdystrophins, mini dystrophins, and our utrophin, this is the transgene product, utrophin and dystrophin, but we see this fragmentation product, especially under mechanical loading. So right now we are limited to AAV size, we've been trying to improve the stability by increasing the tensile strength of the shortened transgene product so it can kind of function like the full length. And in our design, okay, there's a Western blot right here of our nanotrophin next generation, and the band is totally gone. Imagine. Oh, yes.
in the last data set that you showed, there were 2 animals that had increased fibrosis limited expression. Why like that?
That's a great question. I looked at vector copy number and mRNA. So we have vector copy number, which are vector genomes per diploid genomes in the pink, and then relative mRNA in the purple. And these are each individual microeutrophin treated mice. And these are the two animals that had the low expression, which is really weird because I detected 15 in this one, and this was done in triplicate. I even did it twice because this was a little weird to me. And then this one had below average on both. So this mice makes more sense, except that there are other mice that are close to it that were able to express more. I do think our Western blot is maybe diluted out with all that fibrosis. So when we load total protein, we're not getting all just myocardium proteins. We're also getting the fibrotic collagen proteins.
It's the one and done, right? And so like
Yeah.
20% or more patients can pull this. But the genetics of the mouse or something.
Yeah, that's a really good point because I've thought about this so much, and I'm like, it clearly the resistant myofibers that have all of the vector genomes are the ones producing all the utrophin shown in this graph, which means all of that, the myocardium or the myocytes that maybe had a lower vector genome that didn't produce enough microutrophin, they weren't protective and they died off and were replaced by fibrosis. And so now you just have these resilient myofibers. But I don't know that mechanism of why, so it's been really interesting.
Other questions? Let me take this opportunity. to
There's
invite-
one.
Other question.
So you're using utrophin instead of dystrophin. I know that dystrophin binds microtubules with the heart, so
Mm-hmm.
so that these models cases.
That is a great question. What we are seeing is yes, but I have not actually looked at any readouts to actually compare that with wild type to that level. But that's such a great point. They don't have microtubule binding sites. Yeah.
If there are no other questions, I want to invite everyone here to come up to the Feller Chance 7 room at 4:30. I think there's enough time here. And we'll have some libations, some of the Quill's favorite foods.
Cool. Can I do my extended acknowledgements real quick? Be like 2 minutes. Okay. All right. This was right after I passed my prelim, and I think he's drinking out of a beaker. And we're all masked up, but it has been awesome working in your lab. I joined it when it was 2 grad students, Leon and Chris. It was Leon, Chris, Hansel, and Ben. It was literally called the Bro Lab. And I couldn't have picked a better lab. It was such a great fit for me, and I've had a lot of fun, great mentorship. Next, I want to actually thank my lab and Robin, honorary member. It has been such a dynamic lab over the last few years, so it's just been great to have such a good support system. Next, my class, my cohort, 6th years, I think it's me and you, we're left. But you made this time unreal, thank you. And of course, all of you, GTV, amazing. Then I wanna hit on my Philly rower friends, just my escape from lab. Also my team, my teammates, and a few are here. And then of course, the boys, all of them are my rock, essentially. And then we have my family, and a lot of them showed up. So my mom, my aunts right here. Next, I have my fiance, which is really weird to say because it only happened like 3 weeks ago. But it's been awesome. This was before we were even together. We were friends for 2 years. So it's just, he's seen me this entire process. It's been really, really good support. Next, I have my sister. She's out in California. That's where I'm from. She couldn't be here because she's working a lot. Okay, real quick. She majored in writing and mastered in writing, and then she started working for Illumina as a technical writer. During the pandemic, she taught herself how to code, became a web developer, and then she got promoted to senior software engineer at Illumina. Huge, so cool. Okay. I'm not gonna say much here, but my dad, I lost my dad 3 years ago unexpectedly. So that was really hard and right in the middle of my PhD, but he was the one that really pushed me to do the PhD. He worked in construction and then he started to go back to school when I was in college. We were both studying biology at the same time. And so right here, I ended up doing a lab tech position for Gary Owens at UVA. And he was like, this lab is gonna lead you to a PhD. And hopefully by the end of today, it did. And then lastly, my mom, she's gonna cry. She's right here. She's been so supportive for just anything that I'd ever wanted to do and always showed up for me. She always flies out to Philly. We always go on really cool trips. And so I'm just really grateful for her. So thank you.
Hope to see some or both of you at 4:30ish. I'm not gonna win. Something. Awesome. Go crush it. Your home's left. See what's popping out.